Assalamu alaikum and welcome back to our course DNA and Design. So now that we have gotten a background on DNA, we would like to get to where we wanted to go, which is the evidence that DNA provides for design. And that goes hand in hand with the evidence against a random chance process producing the structure. And we will cover several different topics in this regard. But I'd like to begin with a quote attributed to Charles Darwin from one of his letters, where he says, yet he began by saying that he can't dismiss the universe as haphazard, but he says, yet I can see no evidence of beneficent design, or indeed of any design of any kind in the details. Of course, Charles Darwin did not know about DNA. It was discovered significantly well after he lived and died. And our job here is now to begin to look at the details, because when he looked at the details, they were at a much coarser level than us now being able to look at the details. And so with that, inshallah, we begin. I'll just remind you very quickly that we said that the DNA is a double helix, two complementary strands made up of a four-letter code, which are the DNA bases, T, A, C, and G, and that they are transcribed to RNA, and that four-letter alphabet gets divided into three-letter words known as codons that specify the amino acids. So the order of the DNA bases affects the order of the RNA bases, and the four-letter alphabet is used to make three-letter words or codons, each of which specifies a particular amino acid, and all of this is going toward making a protein. Once the amino acids are all linked up, they will make a long protein chain, which then folds into a complex three-dimensional shape to make a functional protein. We're reviewing this material because we will also have recourse to it later on in the lectures. But we can think of the main function of DNA rather, is to make proteins and to do so via the intermediary RNA. Because proteins are the machines of life. They do the essential life work and build the essential life structures. So we have that DNA is a code. It is a code for transmitting information on how to build proteins using its four-letter alphabet arranged in three-letter codons, as we have reviewed. Now, just knowing that it is a code to transmit information allows us to think about things like the efficiency of a code. Whenever you have a code, you can ask, well, how efficient, how good is this code at resisting the effects of error? And that is really the topic of this lecture, the efficiency of the DNA code, which I think is the first and most compelling piece of evidence that this code was designed and did not arise by chance. To get into thinking about this idea of efficiency of a code, let's think about computer codes. We're all familiar now with the fact that computers use ones and zeros, bits in the computer, so-called, to transmit all the information. And so let us look at the code that we want to transmit on top. The code that we want to transmit is 1001011. Now, electronic noise, chance errors, and so forth can flip at random any one of those bits, and we end up transmitting the wrong code. So, for example, when you look on the bottom, let's focus on that first bit, which is a 1 in the code that we want to transmit. If it by chance flips and becomes a 0, then instead of transmitting 1001011, we would transmit 0001011. So we could think of that mistake as a point mutation in the code, just like we talked about point mutations in DNA. So how do we try to avoid such errors, or how do we try to correct for such errors? Well, in computer science, there are very complex algorithms to do this. If you've studied computer science, you know, for example, about Hamming codes, named after Richard Hamming from Bell Labs. But for our purposes, let's just think about a very, very simple approach. Let's say we're going to use a 3-bit correction algorithm. What does that mean? It means that whenever I want to transmit to you a 1, I'm going to instead send 3 ones. And when I want to transmit to you a 0, I will instead send to you 3 zeros, and we will use a majority rules approach, and we'll explain what that means. For argument's sake, let's say that I have a 10% chance of error. I have a 10% chance that any bit could flip and change from 1 to 0 or 0 to 1. So if I focus on that first bit, which is a 1 in the code I want to transmit, I'm going to instead send you 111. Now that whole code on top would become, instead of 1001011, would become 11100000111 and so forth, because I've replaced each one with 3 ones and each 0 with 3 zeros. Now how can that help? Well, let's again think about that first bit, the 1 that I want to send you. If I know that I want to send you a 1, I'm going to send you 111. So what can happen to that message? Well, that 111 could come to you as intended as 111. Or I could have any one of those 3 bits change. And so I could instead send you 110, because even though I intended to send 111, that last bit flipped to a 0. Or 101, or it could become 011, and so on for the list of 8 possibilities on your right. But now here's the important part. Remember, each bit has a 90% chance of being transmitted correctly and only a 10% chance of being transmitted incorrectly. So those 8 possibilities are not all equally likely. And if you get 111, you will assume that I meant to send a 1. And we said majority rules. So if you get 110, you will assume I meant to send a 1. If you get 101, you'll assume I meant to send a 1, and so forth. Now let's say you end up getting 100 because 2 bits flipped. In that case, you will assume I meant to send a 0 when I really meant to send a 1, and that would be a mistake. But how likely is that mistake? Well, remember, chances are, if I want to send 111, you will get 111. How likely is it to get 100 instead? Well, it's 0.9, 90% chance that the first one will be transmitted correctly, and then a 0.1 that the second one would become a 0, then a 0.1 that the third one would become a 0, and that works out to 0.009. So there's only a less than 1% chance that you would end up getting 100, and the same analysis applies for 010, the same analysis applies for 001, and for you to end up getting 000 when I meant to send 111, that's a 0.1% chance. So if I use this 3-bit correction algorithm, and if I've done my math correctly, then for a 10% error rate, the 3-bit code increases accurate transmission from 90% to over 97% per bit. Now, this is a very poor way to improve correctness. It is less error prone, but it's very poor in efficiency, because now I need to send 3 times as many bits as my original message contains, but you get the idea. that we can mathematically analyze efficiency, and we want to do that for the DNA code because it's very important for DNA to be able to accurately transmit information on how to build a protein. For example, I want to be able to build hemoglobin correctly so it can carry oxygen efficiently. And so our question will become, how good is DNA at transmitting the necessary information to build a functional protein in the face of point mutations? How efficient a code is the DNA code? And when we look at DNA, we find an amazing surprise. And this sort of thinking and this sort of analytic paradigm is really rather recent. It's perhaps beginning in the early 90s. And what researchers have found is that DNA has exceptional error minimization capacities. The DNA code is a wonderfully efficient code. So when we look at the DNA code, let us go back to the slide that we put up previously, because what that code is a way to assign 3-letter codons to particular amino acids. And it turns out that we can analyze in a bunch of different ways how good a code this is. So let's look at this table. And you see that the table is sort of divided into blocks of different colors. So for example, leucine is coded for by 6 different codons, UUA, UUG, CUU, CUC, and so forth. Now, if I want to look at changing this genetic code, there are a lot of different possibilities to do that. One easy possibility to sort of think about is if I keep the block structure of the table the way it is, but I put in the names of the amino acids at random. So for example, in the first block, which is 2 codons, UUU, UUC for phenylalanine, I simply randomly shuffle the 20 amino acids. I keep the stop codons the same. And so maybe, for example, phenylalanine would be changed to arginine. If I did that, how many possible different codes than the true primordial genetic code than what we call the standard genetic code would I have? It turns out it's actually a staggeringly large number. It's about 2.4 times 10 to the 18th. So 2.4 billion different codes. So how good is the standard genetic code compared to those 2.4 billion possibilities? That is a question that we can ask. Now, before we go on to look at the answers to this question, I'm just putting up a blurb from a recent research article here. And the point I want to make is that when we said there are 2.4 billion, that was keeping the block structure of the table the same, but shuffling the names of the amino acids. But I don't have to keep the block structure the same. I don't have to say that I'm going to have a block of 2 followed by a block of 6, etc. What if I consider that I'm going to have 3 stop codons, like the standard code, and then I have 61 codons, and I want them to somehow encode for 20 amino acids, the 20 amino acids that make a protein, with the only condition being that at least one codon should code for each amino acid. In that case, there's a staggeringly large number of possible codes. It's actually about 1.5 times 10 to the 84. That is an unimaginably large number. It substantially exceeds the number of hydrogen atoms in the entire universe. So the number of possible codes that 3-letter codons coding for 20 amino acids could have been is more than the number of hydrogen atoms in the entire universe. So when we ask, okay, then how special is our genetic code in comparison to all of these possibilities, we actually get a fairly stunning answer.